Welcome back, everyone. So for this week, we're going to be doing our induction and expression of our beta-gal gene. So in this flask, you can see that I have about a liter of media that has been inoculated with our colony, as well as I've added ampicillin. So I let this go on for I let this mix for about 6 hours. And now it's reached an optical density of about 0.6 on the spectrophotometer. So now that it's reached 0.6, what I want to do is add 1 ml of 100 millimolar IPTG. So this should begin our induction. All right. So with that, I'm just going to throw it back in the incubator so it can induce for another 3 hours. So our cells have finally been induced for about 3 hours. So now the next thing I'm going to do is I'm going to split this 1 liter of our cells into two 500 ml volumes and spin them down in a centrifuge at 6,000 Gs for 30 minutes. This way, that after these 30 minutes, I expect to have a pellet that I'll be able to resuspend and I guess work with to continue the lysis process. All right, so now we finished centrifuging the cells and we're able to get a pellet. So here you can see is one of the pellets that we got, and each of these pellets weighed about 2.5 grams. So to each of the pellets, I added about 3 mLs per gram, so altogether 7.5 mLs to each pellet because there were two. So I've already taken the liberty of resuspending one of them, so you can see that after resuspending one of the pellets, we're able to get about 10 mLs of resuspended cells. So what I'm going to do now is just resuspend this one here, and then combine the solutions, and then we can actually begin the lysis. Alright, so I finally finished resuspending the cells, and here you can see we collected about 20 mLs in total of cells. So the next step I'm just going to do is I'm going to add about 20 milligrams of lysozyme. So lysozyme is an enzyme that specifically degrades the peptidoglycan layer in E. coli cells. So hopefully with this lysozyme, we'll be able to break down the peptidoglycan and our cells will be able to burst. So now that I've added it, I'm actually just going to put it on this hula shaker just so it can kind of spin for about an hour, just to let it thoroughly mix into the resuspension. Alright everyone, so it's now the next day, and I just want to let you guys know what I ended up doing last night. So remember, last night I threw this in the hula shaker. I let it spin for about an hour and a half, just at 4 degrees Celsius, just so that the lysozyme could completely mix with the resuspension and slowly break down the peptidoglycan layer. However, that's really not enough to completely lyse our cells. So what I ended up doing next was starting a freeze cell cycle. So the way I do it is I'll just take the cells after they've kind of been mixing with that lysozyme and throw them in the freezer, so at minus 20 degrees Celsius. So you can see that right now the resuspension is completely solidified. We do this so that when we freeze the resuspension, the ice crystals can actually form and puncture the cells, rupturing them. So we do this about 2 to 3 times, just throwing it in the freezer, thawing it out, throwing it back in the freezer, thawing it out. And by the end of it, we should see a very clumpy mess that kind of indicates our cells have been completely lysed. So I'm just going to continue this cell lysis through the freeze cell cycles, and I will get back to you when the entire cycle is done. Alright, so I've now finished my 3rd freeze cell cycle, so I'm pretty sure that now our cells are completely lysed. A good way to know that the cells have been lysed is when you look at the solution, you can see that it doesn't kind of flow as well. The solution has become incredibly viscous, very clumpy. And the reason for that is because we lysed our cells, everything inside the cell kind of moved outside the cell, and this includes the DNA. So all the DNA that was in those cells can kind of act like a net and wrap around all the proteins as well as all of the other, I guess, trash that's in the solution right now. So what we need to do to make the solution go back to being being less viscous is to remove that DNA or chop it all up. So that's what our next step is going to be. So we're going to be adding DNase or deoxyribonuclease, which will cut the DNA. And we'll also need to add magnesium chloride. So magnesium chloride, the only reason it's there is to activate the DNase. Without magnesium chloride, the DNase won't be able to really function properly. It doesn't really have to be magnesium. It can be any divalent metal, but metals like magnesium and calcium tend to work the best when activating DNase. So I've already added the actual DNase into my resuspension or my lysed cells. So I'm going to add 2.22 mLs of my magnesium chloride. So this should make a final concentration of about 100 millimolar in solution. So 1, 2, and then just going to change it to 220 microliters. And then add that. All right, so now that I've added everything, it's a little hard to tell, but you can kind of see at the bottom there lies all of that magnesium chloride. So I'm just going to put it back in the hula shaker for another hour to an hour and a half to let everything mix very well. And we're hoping that after it's done mixing, the solution should become very smooth again. All right, so just kind of jumping into the future. So the last time what I showed you was me putting our lysed cells in the hula shaker after adding DNase so that we could chop up all the DNA. And the reason we did this was so that we could basically make the solution less viscous so that when we spun it down, the DNA wouldn't pull our protein down into the pellet. We want the protein to stay in solution. So after I kind of let it shake on the hula shaker, the solution did actually become very liquidy. It wasn't no longer viscous. And the last thing I added was not magnesium chloride, streptomycin sulfate. So streptomycin sulfate is a salt. I added about 666 milligrams to my lysed cells. And the reason being is that this streptomycin sulfate, what it'll do is it actually will precipitate out the DNA. So this means the DNA will come out of solution. So when we spin down our tube, we're able to get a pellet that's just full of streptomycin sulfate, junk parts of the cell, and the DNA. So that's what ended up happening here. So you can see after I spun it down, we got this really large pellet, and this is all cellular debris, so our DNA plus that streptomycin sulfate. And in another tube, I actually collected the supernatant. We got about 24. So what I'm going to add to this supernatant is a chemical called ammonium sulfate. So that's what this is here. So this is a saturated ammonium sulfate solution. You can tell because at the bottom of this bottle, there are a ton of these crystals to kind of show that the solution can no longer take any more ammonium sulfate. So for all intents and purposes, this solution is 100%. So what I'm going to do right now is I'm going to add about 12 0.67 milliliters of the saturated ammonium sulfate solution to the supernatant that I have collected. And once this ammonium sulfate solution gets mixed in, what will happen is it will actually precipitate our protein. So all the protein that was inside the cell will actually just get precipitated. So just want to make sure I get the right amount. Okay. And so you can see right now it's very clear looking. Once I add the ammonium sulfate, you can see that it starts to get a little bit more opaque until eventually it is completely opaque. So I am just going to I'm just going to put this back on the hula shaker for about 10 to 15 minutes just to let it mix. And then I'm going to do one more centrifugation. And whatever I get from this pellet, that is our actual protein that we want to collect. So let's talk to you guys after I'm done with that. All right, everyone. So we've now finished up with the last centrifugation. And as you can see, we got a really large pellet. So this pellet that you see here is all of the proteins that were made in the BL21 cells. So now the last thing we kind of just need to do is resuspend this pellet and aliquot it and store it, so that sometime in the future we can try purifying our actual protein from all the junk protein that's also in this pellet. So all I'm doing right now is just removing the supernatant. And that's just going to be trash. And what I'm going to do is just add 3 mLs of chromatography buffer. So all chromatography buffer is just buffer that we use for our chromatography systems when we purify our proteins. So I'll talk about this more in the next lab when we actually start purification. But all you need to know is that the reason I'm adding this is so that when we resuspend the proteins, it's basically in an environment that that's very similar to what it's going to be like when it's in the actual chromatography machine. All right. So you can see here I've added the buffer. All I'm going to do is just resuspend this pellet gently, making sure I don't make too many bubbles. I don't want to disturb the proteins now that we finally have them collected. So I guess I'll just do that off camera and then end up storing them in a -80 freezer. But that's pretty much the conclusion for this lab. After you resuspend the pellet and put away the proteins, that's all you really need to do. So I'll see you guys next week when we actually start our first round of purification.